Hi everyone, today I'm going to be sharing with you an osmosis and diffusion lab that I adapted for my students who are doing distance learning. I just want to show you first, this is the original lab packet that I would have given my students. And here's an introduction. I just pretty much converted everything on here into a Google Slides version with links to video clips that I had pre-recorded. Here's the introduction, and there are video notes that students can take when they're watching this really quick time lapse, just answering what do you think is happening here in the video. This is the basic iodine and starch dialysis tubing activity. Then students are going to design an experiment with 5 unknown samples of saline solution that are colored. You can see the 5 unknown samples are over here. This is just an overview. It's a picture that I took of my entire lab setup because I thought it looked really pretty with all the colors. And so students can only differentiate these solutions by color in the beginning of the lab. Students are tasked with brainstorming and designing an experiment using only the materials provided here. I gave them some of these materials are here, and students are going to determine. So the task is for students to brainstorm and design an experiment using only these unknown solutions: a scale, weigh boats, and various vegetable slices. I have students select only one. You can have them select 2 and then eventually make some sort of comparison. And you can also change up how you want, like what kind of question you want your students to be answering. I gave them a more straightforward question because I modified this a little bit and I realized my students just needed that structure a little bit more. And so my main task is for them to determine the molarity of these unknown samples, using what they know about osmosis, the movement of water in and out of cells, and applying that to these plant cells, these different plant cells. And that will be followed by a determination of the saline concentration of their specific vegetable of choice. And I'll show you how they're going to do that later. So number 3, you can take this out if you don't want to be this specific. I added this later for my students because they just needed that structure. So all the adaptations that you see here, this is like after I have done this with my students. So I made a few adjustments, and you can make your copy of this entire slideshow and make your own adjustments as you see fit for your students. I know that it's supposed to be about inquiry, and so I did go through that process with them. I just updated the slides with more guidance to help them in that process because this is their first ever AP Bio, semi-lab, since they're not even doing this lab themselves. And I had them also list out what I also had them list out what their independent variable dependent variable constants, controls, if any. They were to brainstorm this in partners or as a group or individually. I have very different class sizes between the 2 classes that I teach, so I just did whatever fit the vibe of my class. Some students wanted to work in breakout rooms with friends, others wanted to just be in the main room and have a discussion with me. So it was very flexible. And I just have this note here for students to know that they're not conducting this lab themselves, even though I want them to think about and brainstorm and design an experiment. They can put this in part of their lab write-up, like what they would have designed. And then when they watch my clips, they will have to write out what they see in my procedure. And I just have 3 video clips. I'll open each one of these to show you what they look like. And yeah, that's pretty much the instructions I gave my students. And I have this slide here, but you can choose to move this slide after you go over the rest of this lab report. Students have to include qualitative and quantitative data for this lab report. They have to show all mathematical calculations with proper units, organize a table to present their data, especially with the initial and final mass of the vegetable after having it soak in the solution for an hour. They also have to make a proper graph, and I'll show you what that looks like because I made a sample for my students as well. And in their conclusion paragraph, they have to include the following. I also have a rubric and lab template, a lab write-up template right here that I made available for my students. Most of them decided to copy and paste, or not copy and paste, make a copy of this and then just delete my instructions and fill in these blanks so they have like this structure already there, sort of like a skeleton for them to use. And this is my lab rubric, also linked in the slides. And the discussion part has 3 sections: a conclusion, experimental analysis, and overall reflection. All of that is, so the details for all of this is here. Some of it is also right here for students to reference. So I have a lot of reminders for students on what they have to include in their final lab write-up. Now coming back here, so the highlighted parts are the parts that I tell students to double check in the rubric as well as the template. And then this last slide here, this is just questions for thought that I copied and pasted from my adventures on Google trying to find some relevant guiding questions for my students for this lab. I just didn't get to actually using these questions, but they're good for discussion. You can also have students answer them in their lab notebook, in their lab write-up, or just as a sort of review as a whole class discussion after the lab, or maybe pre-lab activities, anything you want. I just have this here on this slide. And for my students, I just had these here for them to think about, and they can discuss some of these in their discussion and reflection as well. So again, this is my 2nd year teaching. This is my first time making something like this. I just adapted my materials from this lab that I got from another teacher. So I hope this works for you. If it doesn't, I hope you find a better way to do this Osmosis Diffusion Lab. But this is a resource that I wanted to make available for everyone in case you don't have time to set everything up and record your own clips. This took me almost like 4 hours, maybe, yeah, I think a total of 4 hours. Just the whole setup and then getting all my materials and then waiting and then weighing everything, and just the whole setup took way longer than I had expected. So I'm hoping someone else can also benefit from all the clips I took, and I'll show you what each of them look like now. Before I show you, I have this shared, no, not this one. So I have this entire album of a few pictures and also all the videos that I have linked on the slides. This is just an album I have, and it's just easier for me to see everything at once since the links only take you to specific videos. So each of these links, the first one I just opened, introduction and setup and materials. This one students can view and just see what my basic setup was, and they can use this to fill out their material section of the lab report. So I told my students to imagine that they were me and they were the ones doing this lab, and everything I did, they did. So I just wanted them to pretend that they were the one doing it. Here I have my videos for the general procedural steps. The first video only shows one vegetable, and it actually shows, it's like 55 seconds showing me cutting the vegetables, so it's the same process for all the vegetables, you cut them. And the second video shows me weighing each of the slices and writing down how much they weigh. All of their initial mass I put down and then taped onto a weigh boat just so I could keep track of everything. And then the third video is me basically pouring, mixing first all the solutions and then pouring all of the colored solutions across. And I told students to only focus on one vegetable. You can have them focus on two. The setup looked like this because it was easier for me to just, go down the rows and fill the whey boats with liquid. This last one that you see here, the graduated beaker, contains tap water, and that's the control. So again, you can have these discussions with your class as a class, or have them discuss in groups and then bring them back, or you can guide them through the process. And let me show you what the russet potato looks like. This is my first initial setup, so this is the one that looks really weird. Here I'm showing the different masses of the different slices. So if I click on this, you can actually watch the video. And I just go, I guess, up the column. And you can see the different colored solutions, the vegetable slice, the potato slice, how much it weighs. Students are going to take notes. So the notes that they take can be in their notebook, or they can take notes directly in their lab write-up, and they just have to organize everything. So I would tell my students to take notes on the colored solution that the potato slice is in, as well as the mass of the potato slice in that colored solution. So this is the initial mass, and I'm going to close that and bring. Now here are the final masses, and there are 3 videos because I sort of messed up, and you'll see here. So initially this took forever. I was going to dump out the water and then I was going to dump out the solution and then weigh each of the potato slices. This took forever. It was like dark outside by the time I started doing this, so I ended up finding a quicker, more efficient way to do this in the other videos, so that's why this is a little different. I tap the vegetable slices dry and then I weigh them. Students will look at the weigh scale to get the notes for how much it weighs, the final mass of the vegetable slice. And I did that for all of these slices, only to realize that I wanted students to gather qualitative data as well. And for that, I made another video retroactively, which was not the best, because now it's kind of hard to see which potato slice came from which solution. You can tell like the pink and purple ones probably, but the others, maybe ones like light green. So it's up to you if you want students to use this clip or this potato. So it's up to you if you want students to use the russet potato because of the way I sort of messed up in making my video clips. I still gave this to my students and they were able to glean whatever data they could from this for the qualitative, and my students were able to glean whatever qualitative data they could. I told them it wasn't going to be a big deal because they would have the calculations from the before and after masses, and that was all that mattered. And then I have videos for all the other vegetables. Again, the before, which looks like this. I just go down the row and students will take notes of the color of the solution and the mass of the vegetable slice. Video 2 now is a lot better now that it was my second time recording and I knew exactly what I wanted my students to see. And yeah, so this was my setup second time around, which is a lot better. You can see me dabbing the potato slice and then trying to bend it so students can gather qualitative data as well, and then the quantitative data from the weigh scale. So this is how students are going to collect their qualitative and quantitative data, which they should make 2 tables for, or actually I gave my students the freedom of creating whatever data table they felt they needed to present their qualitative and quantitative data, as well as their calculations and their final presentation of the percent change in mass, as well as a proper graph. Okay, so now I'm going to show you what that proper graph should look like. So students will calculate their percent change in vegetable mass by subtracting the initial mass from the final mass, dividing that value over the initial mass, and then multiply by 100. That would be the percent change in mass. And here are my 5 different colored solutions and my control. My x and y axis would be, the x would be solutions of varying saline concentrations. The y-axis would be the percent change in mass. And I would like students to organize this in increasing percent change in mass, which they can do by highlighting, I believe they have to highlight everything, and then customize sort sheet from A to Z would make it in increasing percent change in mass from negative to positive, which is what you want. And I just need to, whoops. Now I'm just gonna rearrange this because I don't know why. Okay, so I just rearranged this so that the labels are on top. I'll highlight everything and then I'll insert a chart. So this is the chart that is going to show up. It's the most simple line chart, but you don't want that. You want this column chart. And I'm also going to customize it a little bit more. This is what I expect from my students. I want them to put a trend line. This trend line is going to be important for determining the approximate saline concentration of the vegetable slice. And I also want data labels for each of the percent change in mass. And then I will go to grid lines and ticks. So I just like seeing more specific values. I don't like there being a lot of white space. So for the vertical axis between 0 and 0.2, if I add one minor spacing line, then I know that this one is 0.1 and 0.3. So I just prefer that. It's just a personal preference. And then let's see what the x-axis. The x-axis isn't really going to do anything. It's just like down here, so they don't have to do that. And so when this is organized in this way, you can see that if there was a large negative percent in mass, you can determine that the green solution is probably the most hypertonic solution, meaning it has the greatest molarity of saline concentration, which results in a significant decrease in mass of the potato or whatever vegetable slice. So actually, I realized of vegetable, they should indicate which vegetable they are using, as well as label the title, which would be percent change in mass of whatever vegetable they have in different colored saline solutions. Okay, so the green solution would be the most hypertonic, meaning that the vegetable slice ended up losing all of its the vegetable slice ended up losing a lot of its water to its surroundings, which is more concentrated than the inside. And here on the very far end is, let's look at the red, actually the control. So the control is just tap water. So you can see the control had a significant increase in mass for that vegetable slice, and you can determine that this is because the control, being tap water, it is hypotonic to the vegetable. So the vegetable has more substances, more saline concentrations that resulted in the water diffusing into the plant cells of that vegetable. And you'll see the best fit line here. It's going to come into play now. It intersects this line where it says 0% change in mass. This would be where the saline concentration outside the vegetable is the same as the inside, meaning that water is still moving in and out of the plant cells. However, there's an equal amount of movement on from both sides, and this creates an isotonic environment for the vegetable slice. And you don't really see that here because there is 0.1% change in the mass of that vegetable, and this line actually intersects what would be between the red solution and the yellow solution's molarities. So if we were to apply the molarities, I'm just going to type it out here. Can I? No. Let me just type them out here. So the most concentrated saline solution is 0.8 molarity, and then it's 0.6. I'm just typing it out so that it shows up on the graph, and that will help us determine what it'll help us determine what the isotonic solution should be. So that is 0 molarity, and the control obviously is just tap water, so also 0. So let's say that this is the point marking the 0.2 saline solution, and just right in between this bar, this would be the 0.0. I know this is a very weird way to go about this, but let's do it this way. So you can see that, let's connect this as well. Alright, so this looks really dumb, I know. But okay, so this is 0.2. This area here is 0. This right here would be 0.1 molarity. So you know that the saline concentration of your vegetable slice lies between 0.1 and 0.2 molarity. So students can describe the range in which the saline concentration of their vegetable would fall in, between 0.1 and 0.2. And that's as detailed as they would have to get. So that would be answering this part. What is the salt concentration of the vegetable? They can just give a range. And yeah, I will have this linked in the description box along with my lab rubric and lab template, and also this Osmosis and Diffusion Lab sample data chart that is also linked in here. But I will have all of these linked in the description box, so you can access them, make a copy for yourself, edit whatever you want, and hopefully this is a decent alternative to doing the Osmosis Diffusion Potato Lab in class. I hope you found this helpful, and let me know in the comments if there's anything else I should have mentioned, or if you have any questions, or if you have any suggestions for how I could have improved on this. I'm so open to suggestions because I'm just, learning along the way, and this was the best I could do with time I had. And yeah, this turned out pretty decent. I have not read all of their lab reports yet. Most of their graphs are probably not going to be what I expected, but I'll go over everything in class, so we'll be on the same page. Yeah, and that's gonna conclude this lovely osmosis and diffusion lab. This is one of my favorite labs just because I like all the colors involved. And yeah, I will see you guys next time. I hope you found this helpful.